serves as an electron carrier, and that's going to be important for the Calvin cycle. So that NADPH is required to go over to make the reactions of the Calvin cycle work. Okay, so now let's take a minute there. So we've talked about all these electrons. We've talked about where the electrons have gone, how they've left their photosystems, and they've traveled down the electron transport chains. Okay, but we also were mentioning that this hydrogen was creating a concentration gradient. And as the electron went through this first electron transport chain here, it was providing energy to make ATP. So let's back up for a second and take a look at how that is going to work. Okay, so just a quick reminder here of what we're trying to make. Okay, so here's our ATP. Remember, ATP is made of an adenine molecule, adenine nitrogen base, okay, a ribose sugar, and 3 phosphate groups. Okay, and so ATP is able to do cellular work. It is considered an unstable molecule because of these 3 phosphate groups at the end here. You'll notice each one of these has a negative charge. Remember, negative and negative, they repel. And so this molecule is not very stable because these phosphate groups, they want to break off from one another. They don't want to be attracted and sitting there next to these other negative groups. Okay? And so the ATP is used to help do cellular energy because we will take a phosphate group from the ATP, okay, add it to a molecule, more often than not a protein. So a kinase will take that phosphate molecule, add it to a protein. Okay, remember that's called phosphorylation. So the kinase will phosphorylate that enzyme, which as a general rule will activate the molecule now that it's gained a phosphate group. And so that's how the ATP helps us to do energy. Okay, it helps us do cellular work. So for photosynthesis to happen, the plant has to make some ATP. ATP and NADPH are both required for the Calvin cycle. So these light reactions are going to help make the ATP and the NADPH. And they're going to help do it by creating this concentration gradient. Okay, so we're going to be talking about what's called chemiosmosis. Okay, so think about the word. Okay, and what you know what's happening here. So this is going to be a passive process. Okay, remember osmosis, we're moving things from high to low. It doesn't require any energy. So this is a passive process. But instead of moving water here, okay, I'm moving ions. Okay, so I'm moving ions, okay, across a selectively permeable membrane, and they're going down their electrochemical gradient. So their charge, because remember ions have a charge, their charge is coming into play here. Okay. So chemiosmosis is passive, and I'm moving these ions down, so from high to low, down their electrochemical gradient. Okay. All right, so I've got my membrane here. Okay, here's my thylakoid membrane. Remember, I am zoomed in here. I am way in this chloroplast. Okay, I am looking at those stacks, those granostacks, this thylakoid membrane here. Okay, so if y'all remember your, let's see, the thylakoid membrane, let's go ahead and do this actually. That thylakoid membrane forms, remember it's forming the disc, right? Okay, so I've got my stroma. Remember your stroma with an R is the liquid portion of the chloroplast, kind of like the cytoplasm of the chloroplast. So that's going to be outside my stacks, right? And so this would be what's called my thylakoid space. So I'm inside that thylakoid disc, okay? And so there's my thylakoid membrane there. So what I'm going to do with this to help this chemiosmosis make ATP here is I'm going to pump hydrogen ions. So I'm going to take hydrogen ions that are out here in the stroma, okay, and I'm going to pump them here into the thylakoid space. Okay, so I'm taking these hydrogen ions and I'm pumping them across the membrane into the thylakoid space. Well, intuitively, hopefully, we realize that pumping would require energy. Well, the energy to pump these hydrogen ions across the thylakoid membrane is going to come from when that electron was traveling down the electron transport chain. Remember when it was moving from photosystem 2 to photosystem 1, and we said that as it traveled down the electron transport chain, it was releasing energy. So it was releasing energy to power this, to help pump the hydrogen. And where did the hydrogen come from? Well, the hydrogen came from that splitting of water, right? So to help pump this hydrogen that came from the water splitting into the thylakoid space. Okay, I want to build a concentration of hydrogens now inside my thylakoid space. So I'm creating on purpose a concentration gradient of all these hydrogen ions in the thylakoid space. Okay, so I'm on purpose doing this. So when all the hydrogens, as the hydrogens get pumped into that thylakoid space and I increase the concentration gradient, that's going to represent, this concentration gradient represents potential energy. Okay, having that concentration gradient is a representation of potential energy. And so now that I've built this concentration gradient, the hydrogen ions naturally are going to want to go back, okay, across the membrane. They're going to want to go back into the stroma. And when they diffuse into the stroma, they diffuse through, you know, I'm a great artist, right? They're going to diffuse through this molecule here. And that molecule there is called ATP synthase. Well, hopefully its name tells you what it does. Okay, ACE, it's an enzyme. Synth, it's going to make something. And it's going to make some ATP. And so as these hydrogen ions now diffuse through that ATP synthase, it makes the ATP synthase spin. Okay, it'll spin around like a propeller. Okay, and as that ATP synthase is spinning around like a propeller, it's going to join ADP and phosphates together, okay, to make ATP. Okay, and that's what that's going to be called is a proton motive force. And that's that hydrogen coming through that gradient, coming through that ATP synthase, causing the ADP and the phosphate to group together. If this thylakoid membrane is damaged, okay, or if it didn't exist, this process wouldn't be able to happen. We have to be able to create that gradient. And so this is our chemiosmosis, right? We create the gradient, and so now that I've created the gradient, these ions will diffuse back through the ATP synthase, and they're going to create ATP. Okay, so let's sum this up real quick. Okay, so what I've got going on here, kind of my big idea, I don't want to lose sight of things. Okay, so I've got light energy, okay, which was used to, light energy is going to be used to excite. So my light energy is used to excite the electrons, okay, and make ATP. Okay, it's going to excite electrons and make ATP using that chemiosmosis process, right? And it's going to make NADPH. Okay, and so by doing that now, that's going to help power the Calvin cycle. Okay, this is going to happen both in and across that thylakoid membrane. Okay, got to have that membrane or this isn't happening, remember, inside the chloroplast. My reactants for this so far, okay, I've used the light and I've used water. Okay, if you think of our big picture here so far, I haven't used carbon dioxide. Okay, and my products that I've made so far with this light reaction, okay, is I've made oxygen, which has left the plant, ATP and NADPH, which are not listed on our equation because they're actually going to be used during the Calvin cycle. Okay, so we've got one last thing to look at here with these light reactions. So let's look at what's called cyclic electron flow, and then we'll be all set with the light reactions. Okay, so before we had non-cyclic electron flow, but we also have cyclic electron flow that is happening in the plant. And so this process is older, evolutionary-wise. Okay, and we know this because bacteria do this. Most photosynthetic bacteria do this, and all photosynthetic eukaryotic organisms do this. Okay, so it is considered to be older than the non-cyclic electron flow because bacteria don't do non-cyclic electron flow. That's only in eukaryotes. Okay, so cyclic electron flow only makes ATP. Okay, so it makes ATP, which is great. Okay, it does not make NADPH. So it is not producing NADPH. None of that's coming out of this cyclic electron flow. So none of this is part of cyclic electron flow. Neither is photosystem 2 part of cyclic electron flow. Just the portion right there in the middle. So with cyclic electron flow then, it's only using photosystem 1. Okay, and so the electron is recycled. Okay, so the light energy excites the electron, but instead of traveling down this electron transport chain here to form NADPH, okay, it jumps into the middle of this electron transport chain between photosystem 1 and photosystem 2. So the electron was, our electron here was excited, okay, and now it's going to be jumped into this electron transport chain here as photosystem 2, as the one that the electron travels from photosystem 2 to get to photosystem 1. Okay, and so if you remember, as the electron travels down this electron transport chain, it ends up back in photosystem 1. So that's what we mean by the electron is recycled. It doesn't have to, obviously we're not splitting water, so this would not release any oxygen. Okay, so there's no oxygen released during cyclic electron flow because there's no water being split. Okay, but it is producing ATP because it's traveling down this electron transport chain right here, which helps, remember, provide that energy to pump the hydrogen ions. And this is going to be helpful and it's going to be useful and we're going to need it because the Calvin cycle actually requires more ATP than it does NADPH. So if both of them are happening, if we have both cyclic and non-cyclic electron flow, then that makes the Calvin cycle kind of help make up the difference. It's going to get the extra ATP it needs and not have excess NADPH. Okay, so that electron will just cycle through. It'll leave photosystem 1 and it will return to photosystem 1. So that's our cyclic form there. Okay, so we're going to leave you with, I would like you guys to create some kind of chart, diagram. I want you to try to compare and contrast non-cyclic and cyclic electron flow, and we'll focus on the light reactions when you guys get back to school. Cells in the leaf.